All right, on this section, we're going to look at the labels so you can begin to set in your memory where all these different structures are. I think it's critical for people learning neuroscience to ultimately have the location of these structures somewhat memorized, and this three-dimensional picture of the limbic system in particular down pat become much easier than when we start looking at imaging studies in future lectures. So let's go through each of these areas that we looked at in the 3D model. So here we see, of course, the dorsolateral prefrontal cortex, the ventrolateral prefrontal cortex, and the orbital frontal cortex. That's pretty easy to memorize. You should be able to add the label if you're asked. Now, when we look at the dorsal or more posterior view, here are the colliculi forming the tegmentum. Of course, these are the superior and the inferior. Now, when we split the brain down the middle, the tegmental area is actually right here. That's superior and that's inferior. And on the other side of the channel connecting the third and fourth ventricle, we have a number of areas where cell bodies exist. The ventral tegmental area, so named because it's ventral to the tegmentum, that is where dopamine neurons project up to the cortex. The red here next to it is the substantia nigra, where dopamine neurons project to the caudate, and we'll be talking about that in great detail. The raphe nuclei is where serotonin neurons live, and the blue is the locus coeruleus, where the norepinephrine cell bodies are found. Coeruleus, in fact, is blue in Latin, so that color wasn't selected at random. Now, we are going to take a closer look at each of these neurotransmitters in a subsequent lecture. Now, again, we're looking at the basal ganglia. You should memorize that this here is the dorsal striatum or head of the caudate, the ventral striatum with the nucleus accumbens within it, the putamen, and the globus pallidus with both the externa and interna. Of course, externa means it's closer to the external side of the head. Again, now from a lateral view, we see the full putamen, the head of the caudate, and the tail of the caudate, with the amygdala kind of perched right there on the end of the tail of the caudate. And now looking at the medial side of the cortex, we can see the ACC. You should also remember, although it's not on the label, that the anterior cingulate cortex is divided into the rostral, which is the part that tucks under the corpus callosum, the dorsal anterior cingulate cortex, which of course is right here, and then the posterior cingulate cortex in the rear. And you can also see very clearly labeled the orbitofrontal cortex and the medial prefrontal cortex. Again, sometimes the medial prefrontal cortex is subdivided into the ventromedial, which is kind of in this area, and the dorsomedial here. Now let's look at the limbic system. Here we see the hippocampus, the amygdala, the white matter coming up to form the fornix. We see the arch of the fornix there clearly. There's the mammillary body, the hypothalamus, and the cingulate cortex. Again, rostral cingulate, dorsal cingulate, and posterior cingulate. That gives you a good sense of how those structures relate to each other. Now, this figure is from the text and lays out specifically the Papez circuit. And we discuss in the book the case of HM. HM was a man with severe epilepsy. During the 1950s, he underwent surgery to remove epileptic foci in his hippocampi bilaterally. Upon recovery, he was intellectually intact. He had no motor deficits. His short-term memory was fine, but he could not create any new long-term memories. So he would see somebody, they would converse, they would introduce themselves, the person would leave, come back, the person would come back 5 minutes later, and HM would not know who he was. He remembered people from his past. If his mother came to visit, he would know it was his mother, but then he would forget the fact that she had just visited. And of course, he was disabled, lived for many years in this state, and has died recently. And at autopsy, the absence of tissue in the hippocampi bilaterally was there. So this raises the issue about what does the hippocampus do. As we'll discuss in detail in subsequent lectures, it's very involved in the formation of memory. And to put it in a summary form right now, at any given moment, all the information that we have from the external world, which is represented by the box labeled number 1, enters via the temporal lobe into the hippocampus. It there undergoes processing. The information then leaves the hippocampus via the fornix to the mammillary body, to the anterior thalamus, and then to the cingulate. And here, through processes that again we'll look at in more detail, it is likely that this circuit involves the comparison of what is currently going on in the external world with events that we have experienced in the past. And this allows us to detect what is novel from what is familiar. And so when HM's hippocampus was damaged bilaterally, he lost this ability to look at the current state of the world and draw from his and create new long-term memories, and draw on those new long-term memories to realize that he had experienced something before. You'll note that the final output there is to the hypothalamus, where emotional reactions can be modulated. Activation of the sympathetic nervous system, the fight or flight reaction, such that if we are then encountering something that is noxious or frightening, we will make the appropriate response. So this circuitry will be important not only in our study of memory, but in our study of anxiety disorders, and in particular PTSD. Now, more is involved in the Papez circuit in these conditions. More is involved than the Papez circuit in these conditions. As I noted, the Habenlua and related structures will also be important. But this gives us our first look at sort of functional neuroanatomy. Now, step 10 in the book is a little more complex. Takes us through the purpose of the basal ganglia, as it were. You probably learned from neuroanatomy that the basal ganglia are important in initiating movement. Well, how exactly does this come about? Well, let's walk through the right side of the diagram, which is the neostriatal or sensory motor circuits of the basal ganglia. So let's imagine at rest, or when some ongoing motor activity is being executed, this circuit is quiet. It's only needed when we initiate a new movement. And that in step 1 there, you can see that there are glutamate circuits that go back and forth between the neocortex and the ventral lateral and ventral anterior thalamic nucleus. Think of these as sort of ongoing when there is a behavior that's currently in progress. When we initiate a new movement, and that's indicated with 2, there's glutamate neurons in the cortex that project to the neostriatum, and then there's various outputs to the neostriatum. And we won't get into the details of right now. That will come in a subsequent lecture. But one is a direct output to the globus pallidus interna, and then back again via 4 to the ventral lateral and ventral anterior thalamic nucleus. Another is through B3B, the indirect pathway, which goes to the globus pallidus externa, then through to the subthalamic nucleus, then back to the globus pallidus interna, and again to the ventrolateral and anterior thalamic nucleus. Now, that seems, why do we go through all that difficulty? Well, you will notice a pathway 5, which is going from the neostriatum to the substantia nigra reticulata. And then the substantia nigra reticulata, which are GABA neurons, then project to the ventrolateral and anterior thalamus. They also project to the pedunculopontine nuclei, which is 6B, and to the superior colliculi, 6A, involved in eye movements. Now here's the key point. The substantia nigra reticulata, those GABA neurons, are tonically active. So if they are active, then that's what keeps the circuit at rest. So there is no movement, as it were, because there's this inhibitory input into the ventrolateral and ventral anterior thalamus, as well as the pedunculopontine nucleus and the superior colliculi. So what exactly happens when these direct and indirect pathways get going? Well, when the neostriatal neurons are GABA themselves, so with the pathway to the substantia nigra reticulata, GABA is being placed on those neurons, and we are inhibiting inhibitory neurons. So the substantia nigra reticulata shuts off and releases the structures in 6A, B, and C from inhibition. Similarly, the globus pallidus interna neurons are GABA. So when the GABA neurons of the striatum stimulate them, they will shut off. And once again, the ventrolateral and ventral anterior thalamic nuclei are released from inhibition. Now, the indirect pathway, those GABA neurons are turning off the globus pallidus externa, so the subthalamic nucleus comes to life. But interestingly, the subthalamic nuclei are glutamate neurons, and those will actually stimulate the globus pallidus interna. So the subthalamic nucleus, as part of the indirect pathway, blocks the direct pathway. So the direct pathway is kind of the go pathway. We want to initiate a movement. The indirect pathway is kind of the brake on that movement. And of course, if people have a stroke where they damage the subthalamic nucleus on one side of the body, they develop hemiballism. That is the disorder where on the opposite side of the bottom, when they go to initiate a movement, they'll get flailing of the arms and the leg, because the motor movement is initiated with far too much vigor because the indirect pathway is damaged. And then finally, the substantia nigra compactica, the dopamine neurons, they stimulate the neostriatum, and we'll go into more detail later. But in essence, the dopamine input sort of acts like the sort of lubricant in this system, enhancing the activity of the neostriatum such that movements are initiated more easily. So dopamine is the go neurotransmitter, if you will. The key here, of course, is if you lose dopamine, such as in Parkinson's disease, people will then develop bradykinesia and the inability to initiate new movements. Now, let's move over to the left side of the diagram here, where we see the limbic striatal circuit. It's much simpler, but we have a similar setup. The ventral tegmental area dopamine neurons release dopamine into the ventral striatum, and our connection is with the limbic cortex or cingulate, and then it flows through the ventral pallidum and the dorsomedial nucleus. Now, if we place in an animal, if we place an electrode in the axon that goes from the ventral tegmental area to the ventral striatum, the animal will push a lever in the cage in order to get an electric jolt in that pathway and get dopamine released into the ventral striatum. Similarly, drugs of abuse such as cocaine or methamphetamine will operate by blocking the reuptake of dopamine, particularly in the ventral striatum or nucleus accumbens. Now, when we experience a reward, good tasting food, very pleasurable activity, we get release of dopamine into the ventral striatum and activation of that area. And this creates that good, or perhaps plays a role in creating that good feeling. So if you go out to a new Chinese restaurant and you go in there and the food is wonderful and you really enjoy it, you say to yourself, I want to come back to this place. Later on, a few weeks later, perhaps you're driving past it and you see the sign and you immediately get the sense, well, I'd like to go back there. Well, now the activity of the ventral striatum has been transferred from consuming the reward itself, that is the good tasting food in the restaurant, to signals that reward might be present, the sign that is there. So this system detects the signals that reward is present and sort of creates or helps create the motivation to seek out that reward. And that obviously will become very relevant when we look at both depressive disorders and with addictive disorders. A great deal more to come with that. Now, I mentioned that the habenula is not extensively discussed in the text, so here I'm going to give you some introductory information that will be valuable in future lectures. So again, here's our stria medullaris, which you can see it from the lateral view with the lateral habenula on the side. There is the medial habenula in the middle, and there's the lateral habenula bilaterally. Well, what does the habenula do? Well, it's easy to overlook the habenula. Why is that? That's because if you look at a typical neuroanatomy textbook with a 2D view, it appears to be tiny. It is right there, kind of anterior to the posterior end of the corpus callosum, and you sort of say, well, what the heck is that? Well, you've got to remember the stria medullaris is coming out here. There's the very end of the stria medullaris feeding into it there, and that's all you can see of the structure when you look at a cross section of the brain. You can better appreciate the habenula in this diagram here. So it turns out that this very small structure has very significant inputs. In green, we can see inputs from the basal ganglia, the globus pallidus, the ventral pallidum, the nucleus accumbens, the reward nucleus, and the hypothalamus. And that green input goes to the lateral habenula. And then from the posterior septum, we'll worry about the septum later, goes to the medial habenula. And you'll notice in green, the outputs of the lateral habenula come to the raphe nucleus, so it controls serotonin, but also to the ventral tegmental area, the area that puts dopamine into the nucleus accumbens. And here, the lateral habenula inhibits the VTA, and this is why we now refer to the habenula as the anti-reward nucleus. It becomes active when adversive signals are around or when reward fails to appear. And so we may have a balance between the reward system, ventral tegmental area to nucleus accumbens, and the anti-reward system, which then completes the circuit, nucleus accumbens to lateral habenula back to ventral tegmental area. Now, we've already talked about the hippocampus to the fornix and the mammillary body, and then of course on to the anterior thalamus, and to complete the Papez circuit. Two other pathways I want you to know are from the septum to the stria medullaris to the habenula, and coming out of the septum will include that input from the nucleus accumbens, and then the other pathway, which I'm going to show you in just a moment, goes from the amygdala to the stria terminalis to the ventromedial nucleus of the hypothalamus, and an important area, the bed nucleus of the stria terminalis, or the BNST. Now, all three of these pathways are going to be critical in our response to threat, anxiety, and aggression, and they are important in mood regulation. Now, what is the stria terminalis? I have not mentioned that before. Well, it's an output pathway from the amygdala, and we can see here in green, it actually runs parallel to the fornix, right below it, not part of the fornix, but running parallel to it. But it comes along and it terminates here in the bed nucleus of the stria terminalis. And it does send a few fibers to the nucleus accumbens. Now, the bed nucleus of the stria terminalis, again, an area that's been important in animal studies, but we're thinking a lot more about it in terms of human studies. One very interesting area is its potential role in sexual dimorphism. So here what we're seeing, this is in the rat brain, but we think this is pretty comparable in the human, is that a whole set of nuclei in this area are different in men and women. And the three that are here are the anterior ventral paraventricular nucleus, part of the hypothalamus, the preoptic area, which we're going to talk about when we discuss bonding and oxytocin. And then finally, here's the bed nucleus of the stria terminalis. As you can see, there are differences in volume and neuron number, as well as neurotransmitter type in each of these three areas. And where this is becoming of particular interest is that in general, people who are transgender tend to have organization of these areas, particularly the bed nucleus of the stria terminalis, which corresponds to their gender that they feel is really them. So while they may have been born XX or XY, they do not have the brain organization in this area that is typical of a cisgendered individual. Now, this work is fairly well developed in humans. As you might imagine, these areas are very difficult to image in humans. So the work in humans is controversial, not only from a technical standpoint, but also from people's views of what you're going to do with the data. People who do not believe in transgenderism, tend to want to feel that these are either learned or chosen behaviors, and thus literature suggesting that there are in fact inborn biological differences they find quite threatening to their model. But then people in the transgender community, may not be totally enthusiastic about these either, because they are sensitive to any idea that this is presented as an abnormality as opposed to just a variant. We need to be aware of these things when we get into these very controversial areas, as we soon will. All right, so that concludes our sort of tour of the neuroanatomy. I hope there are a lot of questions in your mind around, well, great, I know where these structures are, but what do they do? How are they clinically relevant? Rest assured, we're going to get into that without too much delay.